In our short list of organelles that we're going to learn, we're going to start with the plasma membrane. All right, I had to wait for the motorcycle to go by. The cell membrane surrounds the outside of all of the cells. And it's usually made of two layers known as the phospholipid bilayer, if you remember, where we have those phospholipids and those fatty acid tails, and they're turned on top of each other, kind of forming this boundary that regulates water specifically. Now here we have an animal cell right here, and here we have a plant cell, and then here we have a bacteria. And if you're looking at the cellular membrane, you'll notice that on this cell, it's on the outside. And on this guy right here, on the plant cell, it's on the inner part. And on the bacteria, it's also on the inner part. And that is because, remember, the plant cell and these prokaryotes have a cellular wall, which is the rigid structure on the outside. So the cell membrane is special in that it controls what goes in and out of the cell. It's very critical in communicating with other cells and maintaining like a stable internal environment. It's like the fence around your property. It protects it from the outside and only lets things in the gate that need to be there. That regulation of its environment, its internal environment, is what we call homeostasis. So I like to think about it as I'm at home in my safe, fenced-in environment, being regulated and protected by this fence, and so I'm in homeostasis. The cellular membrane is comprised of that phospholipid bilayer, and that phospholipid bilayer is two layers of fats. Fats are phospholipids. Remember, they had a phosphate head and these fatty acid tails. And what do you want, Cheesy? Okay, and if you remember, they're turned invertedly on each other, where the phosphorus is kind of on the outside and these fatty acids are inward. And that is because the hydrophobic, or the part that does not like water, is on the inside, and the hydrophilic, or the water-loving, is on the outside, interacting with water. All right, between cats and phone calls, let's get back to work here. All right, so this cellular membrane is all the way around the cell, and remember, it's kind of doubled, right? So that keeps the water inside of the cell and the water outside of the cell. And this barrier helps regulate what goes in and out of the cell. Now you need water inside of the cell, and you need special kinds of fluids inside of the cell. This needs to be there, but you also don't want your cells just flooding with water, so you want to create this space or barrier that helps regulate this internal environment, again, which we called homeostasis. Now, if you can remember, the head part of this phospholipid is polar. Polar things like water is also polar, will interact with other polar things. Hence the reason that this guy right here likes to interact with water. The tails are these hydrocarbons. They're just carbons attached to hydrogens. These guys are nonpolar. So they don't like to interact with water at all. They interact with other nonpolar things. An example of something you might know that's nonpolar is lipids. The phospholipid bilayer is composed of more than just that phosphate head with fatty acid tails inverted on itself. Remember, there are lots of proteins embedded inside of that bilayer. No, not the day to record, apparently. I can't. Okay, so this phospholipid bilayer has proteins that are embedded inside of it. And there's also carbohydrates embedded inside of it. So it kind of has this like rigid structure. If you think about it, it's kind of almost like rolling on a waterbed type of thing, because it has, these phospholipids, but then there's also protein channels that are kind of like embedded into it so that certain things can, get inside of the cell, and also carbohydrates and cholesterols embedded in it, making it not quite as waterbed-like, but a little bit more rigid. And this whole complex structure is called the fluid mosaic model. And that's its name that it gets because it's made up of many different things, hence mosaic, and it has many freely moving parts that kind of move around, and these proteins will kind of just like move around throughout the membrane. Cytoskeleton are these thread-like tubular fibers like this. They're made up of different proteins, and sometimes we don't always see them in a cell, and usually because it just doesn't make the diagram look pretty. But if you look right here, you'll see these little line squiggly, they look almost like hairs. But they're thread-like fibers, and their job is to give the cell like its shape. And they also move around and they kind of float around inside of the eukaryotes, giving the cell structure support for animal cells. Anybody who doesn't have like a cell wall needs some sort of skeletal structure, and that's what cytoskeletons do. For animal cells that don't have a cell wall, they act like shape and structural support for that cell. Cytoplasm is a jelly-like substance, and it's mostly made up of water inside of every cell, and it holds everything in place. It kind of provides a solution for chemical reactions to take place in, because biological chemical processes do better when they're suspended in a solution. So the space in between all of the organelles is the cytoplasm on the diagrams here of the animal cell, the plant cell, and the prokaryote. Now the nucleus contains the genetic material inside. We're going to refer to our genetic material here as DNA in pink. When DNA appears spread out, we call it chromatin, and when it's condensed and it forms that very, very known shape, kind of looking like this, we call it chromosomes. So chromatin and chromosomes are made up of the same thing. It's just that one of them's more spread out and one of them is in condensed form. This all depends on where the cell's at in its replication process. The nucleus is a structure that is surrounded by a nuclear envelope, also called a nuclear membrane in some different textbooks, and it has pores in it. And those pores, they control what can go in and out of the nucleus. Its job is to protect the DNA, and the DNA controls the activity of the cell. So the nucleus houses the most important information, the genetic material that each cell has. You'll notice from this diagram that what we're calling animal cells and plant cells have nucleuses, and these prokaryotes or bacteria do not. Their DNA is just kind of floating around within its body system. Inside of the nucleus is a structure called the nucleolus, and its basic job is to make rRNA, and ribosomes are comprised of this rRNA. Well, actually about 60% of ribosomes are made up of rRNA, and the other 40% it's made up of proteins. Ribosomes specifically are made up of proteins and rRNA, and they're located at different spots inside of the cells. The rough endoplasmic reticulum is one main location, and they're also floating around the cytoplasm. If you look for the endoplasmic reticulum, and you see little tiny little bumps on it, that's also now called the rough endoplasmic reticulum. And it's ribosomes that are attached to that endoplasmic reticulum, making it look rough. Ribosomes are also located freely moving around the cytoplasm. A ribosome's job is to make proteins. The process of making proteins is called translation. Ribosomes on rough endoplasmic reticulum make proteins to export out of the cell. And ribosomes that are floating around in the cytoplasm, they make proteins to use within the cell. If you'll notice, our animal cell, our plant cell, and our prokaryote all contain ribosomes. Rough endoplasmic reticulum has ribosomes on the surface of it, and if you'll notice in all of the diagrams that have rough ER, it kind of hugs the surface of the nucleus a little bit. So the rough endoplasmic reticulum, its job is to make proteins. It'll then package them for secretion to be moved out of the cell, sending it in vesicles to the Golgi apparatus. The smooth ER, smooth endoplasmic reticulum, doesn't have any ribosomes attached to it. Its job is to make lipids. It also stores calcium ions for the cell. But those lipids that it makes are part of the structures that the cell will use in its membrane. Now the Golgi apparatus is this folded membrane. It gets vesicles of proteins from the endoplasmic reticulum, and these vesicles are like little mini shopping carts. They're packages where it's transporting protein around the cell and eventually out of the cell. So this process of Golgi apparatus, items moving through the Golgi apparatus, it processes and then sorts and ships proteins where they need to go. Golgi apparatus is also really just unique to eukaryotes, like our animal and plant cell here. Lysosomes are specific to just animal cells. So plants don't contain, or bacteria for that matter, don't contain lysosomes. It is an enzyme structure, and its job is to break down dead stuff like food, bacteria, other old parts of that the cell doesn't use anymore. It basically will send items through programmed cell death, even the cell itself. This natural process of programmed cell death is called apoptosis. When studying cells, we often refer to lysosomes as the Lord of Death because it engulfs and digests things that don't need to be there. Lysosomes have a chemical reaction specific to breaking down parts of the cell that are no longer used or foreign invaders as well. There's a very low pH inside of the lysosome, creating an environment that nothing can really survive in.